This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 445, recorded on June 8th, 2017. Hey everybody, I'm Vincent Racaniello, and you're listening to the podcast all about viruses. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get 3 meals free with your first purchase with free shipping by going to blueapron.com/TWiV. Coming to you today from Kansas City, we're at the 14th International Nidovirus Symposium, Nido 2017. It's really a Nido meeting. So I have taken 3 participants in this meeting for your listening pleasure at lunch. It was not easy to pick people because there's so many, over 200 people here. All of you could do a podcast. But invite me back and we can do more in the future. So let me introduce my guests all the way on the left from Georgia State University, Margo Brinton. Welcome.
Thank you.
From the Spanish National Center for Biotechnology, Luis Enjuanes.
Hi, thank you.
Is that a good enough representation of your name?
Yes, perfect.
Thank you very much.
Yeah.
And finally, from Ohio State University, Linda Saif.
Thank you, Vincent.
Welcome to TWiV. Now, the weather here in Kansas City, it's sunny and 27 degrees Celsius, although we wouldn't know that in here because we're inside. I can barely see outside. Now, you can see in the front of the table here, I have some TWiV t-shirts. In fact, I have six with me. I'm giving them all away. And I'm going to throw them out. Now, usually I throw them out into the audience. I don't want to throw them because I don't want to hit your food and get them dirty, right? But I'm going to give 3 out right now. So if somebody wants to come, here you go. There's one. 2. 3. Yeah, that's about the farthest my arm can go. I think I have to get one of those t-shirt guns for the future, to shoot them out. All right, so I thought we would chat with our guests about a little bit about their histories and what they do and what they plan to do. And let's start all the way there with Margo. Where are you from originally, Margo? Where are you from?
You mean where I grew up?
Yeah.
Oh, I grew up in Pennsylvania, near Gettysburg.
Wow. And went to high school there as well?
Yes.
Where'd you go to college?
Went to college at Duke.
And were you a science major at Duke?
I was a zoology major.
That's pretty science, yeah. And then you decided to go to graduate school?
Right. I went to University of Pennsylvania in microbiology.
PhD in microbiology. I know that department well. That was in Philadelphia, right?
Right.
And then you did a postdoc where?
Did a postdoc at University of Minnesota.
Mm-hmm. With whom?
And then I worked for 3 years at Riker Labs, which is a drug company of 3M. And then I went back to Penn as a faculty member, and then eventually moved to Georgia State.
So how long were you at Penn as a faculty member?
12 years.
12 years, okay. Now, where did the interest in science come from? You said you were a zoology major in college. Where did that come from?
I think I was always curious about things, and I grew up on a farm, and I always was interested in how things worked. And I had a very good science teacher in high school. My high school was not accredited because they didn't think it was just a country high school, and they didn't see the need for that. But I had a very good science teacher, and that's, I think, what got me interested in science originally.
Science teachers, yeah, that's always it, isn't it? High school science teachers. So you said you grew up on a farm.
Yes.
What kind of farming was it?
It was a fruit farm, mostly apples.
And did you help?
Yes.
in all the chores and so forth?
We all worked
Yeah.
on the farm.
Sure. And so how long now have you been at Georgia State?
How long?
Long time.
Was there, I went there at the end of '89.
End of the 80s, okay. Luis, where are you from?
Spain.
You think that's all we'll understand, right?
I am from the east of Spain, from Valencia.
Okay.
It's in the east coast. And I study chemistry. But I am an old guy by now. And at that time in my faculty were only basic science, like mathematics, physics, chemistry, organic, inorganic chemistry. But no, let's say, biochemistry, for instance.
Right.
And I always wanted to work on biochemistry,
Right.
life science. So as soon as I finished my chemistry studies, I got a sort of job in the medicine faculty. And I was working by myself. That was not a very good idea. So I decided to move to Madrid. And in Madrid, I started working with viruses. That was great. And I was lucky enough, I moved to a lab in Madrid on molecular biology of viruses. And since then, in Spain, virology was well developed because my bosses, but we had at that time very little viral immunology. So when I got my PhD, my boss told me, what do you plan to do? And looking around in Spain, the only good companies were beer companies. So maybe I should go to be a factory because I don't see other possibilities here. And he told me, well, if you go to the States and learn some viral immunology, I will do anything I can to make you inside the system. And I did it. I came to NIH. NIH was a great place for me. I spent 4 years before in Bethesda and later on at Frederick Cancer Research Center. And I can tell you that was so good for me that I went back and got a permanent position in the National Research Center. And so I am very thankful to NIH. NIH was a very good place because so many scientists running through NIH, so many good seminars there. Therefore, this is an experience I recommend to everyone.
Linda, where are you from?
So I'm from Ohio. I grew up outside of Columbus in the shadow of Ohio State University. I grew up on a small farm where we raised cats, dogs, horses, cows, cattle, and chickens. So I went to a small rural high school like Margo mentioned. And then for my undergraduate, I went to the College of Worcester, a small liberal arts college in Worcester, Ohio. And what really got me excited about science, one of the requirements at the College of Worcester is independent study, which is like a mini master's project. So I did my project in the veterinary virology lab at a branch of the Ohio State University on the Worcester. And I really loved it. And so I've really loved virology ever since. But I actually started my PhD program at Case Western Reserve University in molecular biology. And I started in basic immunology there. So I kind of switched. But I'm very happy for that because I've had a combined career in mucosal immunology and virology. And then I got married, so I had to transfer where my husband was, which was on the Worcester campus of Ohio State University at the ORDC. And I worked with a very famous veterinary virologist, Dr. Ed Bohl. My project was on transmissible gastroenteritis virus, the coronavirus of swine. And it was at that time a devastating disease, just like PED in swine in 2014. So I've worked in coronavirus research my whole career, and met wonderful colleagues here at these nido meetings and early coronavirus meetings. So that's my major.
So you've been at Ohio State pretty much all your career?
Yeah, since I've been on the faculty since 1979.
And so I guess, 79, I guess you've liked it, right?
Well, in two career families, it becomes challenging to find dual positions that both of you love. And we were fortunate there to find that position. Plus, it has the animal resources that I really needed for my research, including germ-free animals. And it's one of the few facilities that has that. So that's kept my husband and me both there. And he was an expert in poultry disease.
Yeah, I understand the two career problem, but nevertheless, you've thrived, so it wasn't such a bad decision.
No, thank you.
So Margo, let's talk a little bit about your science. Can you hear me well enough? Is this loud enough?
I'm sorry?
Can you hear me?
Yes.
Okay. I have to tell you that this 2002 paper that you published has been a staple in my virology course. It's called Positional Cloning of the Murine Flavivirus Resistance Gene, 2002. You remember that paper?
Yes.
And I always cite it as one of the earliest papers showing, a host gene that's involved in controlling virus susceptibility. Do you remember why you got involved in that?
Well, I started that project as a graduate student. And in those initial days, all we could do was just characterize the phenotype. But it's an amazing phenotype because you can inject high levels of lethal encephalitis flaviviruses into the brain of the mice, and they don't show any symptoms at all.
Wow, right in the brain.
Yeah. And so it had been shown that the resistance gene for the flavivirus co-segregated with the gene for rickettsia, and they had mapped it to a chromosome. And then a lab in Australia had used recombination in mice to further map down on that chromosome. So they had a region with a microsatellite that seemed to be a reasonable length to do positional cloning. And I had never done positional cloning, but I thought that would be a way to really try to identify the gene. And this was all before the genome had been sequenced. So there weren't many genes in the gene bank. And we assumed that we would find about 8 genes in the 240 region that we thought that the gene would map to. But in truth, we found 20 genes there, so it was a much more dense gene region. And that was due to the fact that one of the genes, OAS1, had duplicated, so there were 8 versions of that. And then we sequenced each of the genes we found in the resistant and susceptible strains to find out where there were mutations that could affect function. And we found then that one of those genes fit that description, and then we tested that in other strains and found that whenever there was a premature stop codon, you had susceptibility, and whenever you had the full-length protein, you had resistance.
Neat.
And we found a gene that normally acts in the 2,5A RNA cell pathway, but it does not have that activity. And so we're still trying to figure out exactly how it works.
Is there any evidence that these control human susceptibility to infections?
No, there is no real homologue of this in humans. So in humans, the OAS1 protein, there's only one gene. It has splice variants, but all of those are active synthetases.
So I teach a virology course, and I use that as an example of mapping host susceptibility, because it's one of the earliest ones where you could actually identify the gene product. So I wanted to tell you that because maybe you hear your ears ringing when I'm talking about it. I want to ask you about an interesting virus that maybe at this meeting many people have heard of, but we certainly have never mentioned on TWiV, so this will be the first time. Lactate dehydrogenase elevating virus.
Yeah. Yes, that's one of the, that I worked with that as a postdoc, and it's a very interesting mouse virus. It causes persistent infections, but the titers go to 10 to the 10 within 1 day, come back down to about 10 to the 7, and then stay at that level pretty much for the rest of the life of the animal. And in most strains of mice, there is no disease, but there are a few cases, C58 mice, for instance, where if there is immunosuppression and cells in the brainstem get infected, then the body still mounts an immune response to the infected cells and ends up causing encephalitis and demyelinating disease.
So is it still called lactate dehydrogenase elevating virus?
Yes, it is.
LDH3.
Yeah.
Okay. And
that's because there's no plaque assay. So the only way you can titer it is that you inject it into mice, and then you measure the elevation of lactic dehydrogenase 3 days after the infection.
I see.
And the reason that it elevates that is because it grows in a subpopulation of macrophages that clear turnover enzymes. And so when they get infected and die, then the clearance is not as efficient.
Does anybody work on this virus anymore, as far as you know?
There was one man in Belgium who kept working on it, and we're actually thinking of working on it again.
Yeah. Let's see if anybody in this room works on LDH3. No. Nobody. Okay. Another interest of your lab over the years has been proteins that interact with the ends of the viral RNA. Right? How did you get interested in that?
Sorry,
So you've been interested in proteins that interact with the ends of the viral RNA.
The 3', 5'.
How did you get interested in that?
Well, we were not the ones who discovered that, but we were interested in the structures at the 3' and 5' end. And so then, as one of our projects, we wanted to see just exactly what you needed to get the 3' and 5' end to interact. So we did a series of mutations down that whole region and found that areas beyond what had been defined as the initial cyclization sequence were also involved in that interaction.
Right.
But as part of that, I was more interested in the terminal stem loops and the cell proteins that they interact with and how those can act as transcription factors for the virus. And we're still pursuing some of those studies as well, now going to structural analysis.
One of the more recent findings from your lab is that components of stress granules interact with the ends, right? Can you tell us a little bit about that?
So the most recent paper was talking about the fact, an unusual finding was that flavivirus infections could resist stress by arsenite treatment, but not by treatments that activated the other kinases that phosphorylate EIF2 alpha, and that would suppress translation. And so we wondered why that was, why there was that specificity. And that led us to a whole study of looking at the ability of these viruses to activate the reactive oxygen species and induce oxidative stress, but then on the other side, a very strong antioxidant response that neutralized the oxidative response and also gave excess capacity so that when you treated with arsenite, there was no effect because the antioxidant pathway was able to control it.
All right. Now, these stress granule components Do you think that their binding to the viral RNA is a means of antagonism, the formation of stress granules?
We think that one of them is involved as a transcription factor for making plus strand, but we don't think that it's being sequestered. Okay. So we don't think that's the mechanism. Initially, that appeared to be the case, but that does not seem to be the mechanism.
I ask you because it's in our textbook that it's possibly a mechanism. Sounds like we have to revise it. So, Margo, if you could point to one experiment in your whole career that you thought was really cool, more than any others.
Oh, that's hard. I don't know, whatever I'm working on, I'm interested in, so I think I can't really answer that.
Okay. I'll come back to you with another hard question.
This episode of TWiV is brought to you by Blue Apron. Not all ingredients are created equal. Fresh, high-quality ingredients make a real difference, so it's important to know where your food comes from. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. They set the highest quality standards for their community of over 150 local farms, fisheries, and ranchers all over the United States. For less than $10 a person per meal, Blue Apron delivers seasonal recipes along with pre-portioned ingredients so you can make delicious home-cooked meals in 40 minutes or less. And by shipping the exact amount that you need for the recipe of each ingredient, they're reducing waste. And the way I like to put it is you can't overeat. Blue Apron's mission is to make everybody a great home cooker. They deliver to 99% of the continental U.S., and there is no weekly commitment, so you only get deliveries when you want them. You can choose from a variety of new recipes each week, or you can let them surprise you. See what comes in the box. Here's some upcoming recipes: warm smoked trout and asparagus salad with fingerling potatoes and garlic crouton. Spiced zucchini enchiladas with creamy lime and tomato rice. Elote-style vegetable tostadas with summer squash, poblano peppers, and cilantro rice. Man, makes you hungry, doesn't it? You can customize your recipes every week, of course, based on your dietary preferences. And Blue Apron's freshness guarantee promises that every ingredient in your delivery arrives ready to cook, or they will make it right. Check out this week's menu and get 3 meals free with your first purchase with free shipping by going to blueapron.com/TWiV. That's blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron, a better way to cook.
Luis, you've worked on a wide range of things. What would you characterize your interest, if you could give it a general description of all the work you've done? What would you call it?
Well, basically, particularly at the beginning, I was interested in viruses infecting macrophages. Because cells from the immune system, I thought that would be relevant. And we have to go back to the old times, otherwise people will not understand this. We got in Spain African swine fever virus. How we got this is because we are near Portugal. Portugal, they have troops in Angola, in Africa. And the soldiers from Portugal, when they were coming back, they brought back food that was contaminated with African swine fever virus. That was a disaster for our country. You know, we produce this fantastic jamon, top quality, so having African swine fever virus will be an economical disaster for our country. So my mentor, Eladio Viñuela, that was training in New York State University. He was just a molecular biologist, but he never in his life was working with mammalian viruses. and he was brave enough to start a new line of research on African swine fever virus, a double-stranded DNA virus. The genome was very big, more than 100,000 nucleotides. So during my PhD thesis, I was characterizing the structure of the genome of this virus and the interaction with macrophages. And young people cannot imagine the work we were doing at that time, because I just want to tell you that a PhD thesis in the 60s was to purify EcoR1. So that was a whole PhD thesis. Very hard, because when you try to purify to really homogeneous protein, you never succeeded. Because when you try to cut a reference DNA, it will be cut in many places. And we got the visit from an American friend, American scientist, expert on restriction endonucleases. He said, oh, come on, don't try to purify EcoR1 to homogeneous state just with the stuff you already have. What you have to do is to look for the conditions under which salt conditions and pH conditions in which only your activity, the activity you are interested in, will be purified. So we were close to suicide, and then with this very simple advice from Bob Yuan, in 1 week we got the activity isolated, just not by getting a better purification, but by looking for the pH and salt conditions that will give us only this activity. So that was really helpful. Sometimes you have to pay attention to visitors that know the things. So we clarified the structure of the DNA of this virus, a huge virus, but things didn't work properly because we thought it was a double-strand DNA virus, but there was some strange behavior. And when we denatured this DNA virus, it turned out to be a single-stranded DNA. So the double-stranded DNA virus had links at both sides, and that became very interesting. Also, it became interesting that many people was claiming that DNA viruses, they don't have too much genetic variability. Certainly that was not the case. They have many huge variability. They lose many genes and completely change the structure. And the other aspect, the interaction with macrophages was very interesting. And of course we were checking at that time whether this virus will infect human beings. Because pig production in Spain was very important and very, very large. And I remember collecting blood from many species, including human beings. And when I infected human macrophages, we realized that they got infected by this porcine virus. We became afraid, but soon later we realized that it was a non-productive infection. But the main problem we had is, I am trying to look for fancy things at that time. There was an assistant professor, a lady at Harvard, and he associated African swine fever virus to AIDS. So she published an article saying that AIDS was caused by African swine fever virus. Tell you the explanation for that. She was an epidemiologist, and at that time in the States, the gays, when they got married for the honeymoon, they went to Hawaii. And the theory how this virus, porcine virus, got into human beings. Because those people in Hawaii, they were eating hamburgers, and when they asked for raw meat, the virus was not completely neutralized. And frequently it's true that gays, they have a stomach ulcer, so the virus from the meat will go in and will cause AIDS. So you can imagine when a professor from Harvard published that, WHO the next day was calling to our lab, and we were very afraid that, I remember I had to purify large amounts of this virus, and they asked whether we could check whether that was true, whether selected sera from people infected with AIDS will react to our virus. We had no P3, P2, P anything. I mean, not to the stage, the standards we have today. And we could prove that there was no relationship between a huge DNA virus growing in the cytoplasm of the cell with an RNA virus growing much smaller one, growing in the nucleus.
So I didn't realize that story. That's very interesting and important to clarify these things, of course. When did you switch to coronaviruses?
Well, first at that time it was obligatory to get a position in Spain to come to the States. I went to NIH, there I was studying with the viral immunology, mechanism of Moloni leukemia virus. I was in a very good laboratory and was very excited. We proposed a mechanism for that. And then I came back to Spain. Even before coming, I was lucky enough to get a permanent position there in the government. And then I started my project on coronaviruses. Since I had experience with virus-macrophage interaction, I had chosen at that time a coronavirus that it was reported to infect macrophages, although that was on the books. The virus I had chosen, only the wild-type virus, the virus straight from the field, they will infect macrophages. But since then, I work with macrophages, many different aspects, until today. At that time, we were nobody until SARS and MERS came in. Mm-hmm. That changed the field completely. Coronavirologists became more important, as you can guess. Okay.
I like to joke with Susan Baker that before SARS, the coronavirus field was a sleepy field. And then all of a sudden it got lots of prominence. Now, I want to point out a paper that you published, which I noticed, being in a different family, engineering the largest RNA virus genome as an infectious bacterial artificial chromosome. You remember that?
Oh, yeah. As you can guess, that was the case, yeah. Many laboratories all over the world were trying to get an infectious DNA clone from coronaviruses. But as all of you know, coronaviruses, they have the largest RNA genome for any known RNA virus, 30,000 nucleotides. And the difficulty of getting an infectious DNA clone is that any time you clone mammalian sequences from either mammalian cells or mammalian viruses in bacteria, they are toxic. So if your problem is getting the infectious DNA clone of an RNA virus with 10,000 nucleotides, that is relatively easy, but 30,000, that was too much. So very good groups in Germany and even in the States, they failed to do so. And always for us, so for Spanish students to be successful, they have to come to the States. And of course, they go back to Christmas time to visit the family. And one of my former PhD students was in the West Coast, California. And, when they came back, they visited us and we interviewed them and I said, "Hey, Luis, the human chromosome has been cloned and sequenced?" And I said, "I don't believe it. I imagine that 95% will be cloned and sequenced, but it's toxic. When you work with those things in bacteria, I am sure they miss the clones with the toxic sequences. So I think they have 98 or so, but not the whole thing. So no, Luis. But in California, they have a special system where they can, a special bacteria and a special plasmids, the bugs. The bugs, and they have the advantage that they grow in bacteria and they only make 1, maximum 2 copies per cell. Therefore, even if the sequence they have cloned there is toxic, they don't die. So, that's very, very interesting. That may help us, because our virus is so large. And I thought that was the beginning of the success. This just conversation with my former PhD student, and the advice they gave me, but when you do the cloning in bugs, don't look for nice, beautiful colonies, only for the tiny ones.
Yep.
Because they grow, but they have problems, so they don't grow too much. So, this advice was great. In addition, we use another strategy. We were working hard, and the strategy we followed to succeed was that we had very well characterized DI genomes. So we had not the full-length genome, but small ones, very well sequenced and characterized. They had deletions, and we were filling in the deletions one by one, and we said, well, that's fantastic, in 2 more months we will have the full-length infectious DNA clone. And we were almost right until we reached one domain in which the PLP domain was highly, highly toxic. But we looked for our private strategy, and we were successful with the cloning. So successful that when they accepted the paper, PNAS, the commentary to the paper was longer than the paper itself. But anyway, that was very helpful because now we can engineer any coronavirus.
So that was the first example of being able to make an infectious DNA.
Yeah, that was the first time.
Copy.
Yes.
Right.
Yes.
Okay. So do you have a particular experiment that you really liked throughout your whole career?
Well, all I know is that lately with the virus-host interaction, viruses, they need the cell to grow up.
They do.
But also to cause pathogenesis. Mm-hmm. So it is very important to do basic studies on virus-host interaction and to identify two aspects. One, which cell genes the virus needs to grow up, because if you identify them, you identify those signaling pathways in which those genes they intervene, then you can isolate drugs that will block the virus growth by blocking those signaling pathways. So we will have antivirals. And the other aspect is that viruses, sometimes they cause pathogenesis not by themselves, but by the immune response they are inducing, and that need the stimulation of specific signaling pathways. And again, by identifying those signaling pathways, then you can identify drugs, and more importantly, you can attenuate those viruses, because if you know the genes involved in this pathogenesis, and you delete those genes from the genome, then the virus becomes attenuated, and unattenuated virus is, in principle, a vaccine candidate. Therefore, I think that we were very lucky when we were deleting genes one by one and identifying those that were involved in virulence. Then we had a strategy to attenuate viruses in vaccine development. And also when we understood some mechanisms of why these viruses were virulent, how they cause pathogenesis, we had another pathway to identify drugs that will block those signaling pathways, and therefore we could identify antivirals. So I think basic research is very important and can lead down to important applications like vaccine development and drug development.
Linda.
Oh, yes, I'm still here.
Yes. I'm sorry there's sound problems, folks, but as I said, you can always listen on Sunday. I know, it's the sound guys. What would you characterize all of your work over the years over? I know you've been interested in diseases of pigs and cows. Would that be mainly it?
And animal models of human disease as well.
So what viruses have mainly interested you?
So over the years, as I said, I started with coronavirus, and we've continued that work in swine and cattle because new coronavirus keep appearing, or they have different tissue tropisms that develop. So I also work on caliciviruses, sapovirus and norovirus. We set up a germ-free pig animal model for human norovirus, and that's been pretty productive. And also the other animal model we set up for human rotavirus. We collaborated with NIH when we established that model to look at the immune factors related to protection as well as how to attenuate the virus, which was the basis finally for human oral vaccines. And then also with TG and our basic research on vaccines and gut-memory connections. So those are the main virus: rotavirus, coronavirus, calicivirus.
So the noro and the rota are to understand human pathogens, right?
Yes, although we've always more or less done parallel research.
Right.
Because rota is also a disease problem in swine, especially just like human infants, baby swine nursing piglets. And when I started working, there were no vaccines for that. Now there are some vaccines to induce maternal immunity, live attenuated oral vaccines. Norovirus, there are some norovirus in cattle that cause severe diarrheal disease, like in humans. There are some in pigs, but they tend to occur in older pigs with less pronounced disease. And then there are sapoviruses that occur in swine that are very similar to the human sapovirus. So swine and cattle have the counterparts of many of these human agents, so we can understand disease both in the host and how this can translate for the human host.
You also mentioned TGEV. Tell us a little bit about that.
So when I started working on transmissible gastroenteritis, coronavirus in swine, we had the scenario in the U.S. and worldwide that was pretty much like what we're experiencing now with PEDV. It came into a seronegative population of swine. There were tremendous deaths in neonatal pigs. It spread throughout the country. There were no vaccines, no antivirals when I started my research. Not that much was known about immune responses in swine. There were no reagents. I had to make all my own swine reagents to study immunology. So that took a lot of time during my master's PhD. But I think the major breakthrough there was to look at the types of immune responses in the swine that recovered from infection in their baby pigs, and then try to develop a way to mimic that type of infection and protection. And we found those were associated mainly with IgA antibodies in the colostrum milk when the pig recovered. If you gave an intramuscular vaccine, it was mainly IgG antibodies and non-protective. So that was the basis for live oral attenuated vaccines for use in sows to stimulate what we found was stimulation of the gut memory link immune system. So we were among the early people to describe the common mucosal immune system in this link between infection of the gut and protection through the milk and colostrum through mucosal secretions.
So is there now a TGEV vaccine?
Yes, there are currently TG vaccines, but what happened was there became a naturally occurring mutant of TG, a respiratory strain that was naturally attenuated. It was more effective in transmission as a respiratory virus, so it spread worldwide, and it's acted as a natural vaccine for TG.
We don't need to vaccinate anymore.
Not so much. Some people do. Sometimes there are some outbreaks of virulent TG in Canada and the U.S., but not nearly as much as there used to be. Also, I think in China.
So the infection is mild enough so that you can let it spread and not worry about pathogenesis?
The PRCV. There was really no attempt to make a vaccine because people thought it was not commercially viable because there was not a disease associated with it.
Now, you've used animal models a lot, obviously, so having a facility where you can do that is important, right?
Yeah, it's essential.
And that's why you've probably stayed at Ohio State. You couldn't do that in New York City.
It would be difficult.
Well, it costs $5 a day per cage per mouse for mice, so I can't imagine what it would be for a pig or a cow. Now, you mentioned notobiotic animals. What's the significance of that?
So the importance of the notobiotic animal is we didn't know to start with what agents was causing what disease. So that allowed us to infect animals that don't have a microbial background at all, no other extraneous viruses. The piglet is such, it doesn't have maternal antibodies. They only acquire it after birth through the colostrum. So that gave us a naive animal and a highly susceptible animal. And so we were able to use that animal to sort out the cause of these various enteric infections. The sapoviruses, the first isolation was in the germ-free pig, the rotavirus, the coronavirus. And what also, in terms of animal models, we were able to model this for human rotavirus, but more importantly, no one could grow human rotavirus strains in cell culture. And of course, that was the basis for the future attenuated vaccines. So we collaborated with NIH. We got some infant stools. We put it into germ-free pigs. It was fully infectious for the germ-free pig. And there are a lot of similarities physiologically, anatomy between pigs and humans, though people don't like to admit it, but there are. So this allowed the virus to propagate to high titer. It got rid of interfering antibodies that were there probably in the original specimen. And then we sent it back to NIH, and we also continued to work on it. That somehow allowed the virus to replicate then in cell culture with the addition of proteolytic enzymes that are present in the gut. The other, I'd say, interesting aspect is that we've continually looked for ways to diagnose these infections, but also replicate these viruses in cell culture. They're fastidious. It's not like MERS and SARS that was so lucky they grew for these respiratory viruses, but these are very fastidious, very hard to grow. So when we worked with sapovirus, it would not grow in cell culture. So what we did, we took the intestinal contents from an uninfected notobiotic pig. We put them in the culture media without knowing what all was there to allow it to go, but assuming it might be something like proteolytic enzymes. And by doing that, we were able to adapt porcine sapovirus to growth in cell culture. So things come around in science very frequently. So what happened is now Dr. S's lab, who's using the human enteroids, was able to adapt some norovirus strains. We found the component in the intestinal contents that was effective was bile acids for the sapovirus. That same component, bile acids, is effective to grow human noroviruses now. Yeah. So it's you never know when some of these serendipitous findings will be applied, to other species, other viruses, and it's been very productive that way, I think.
The serendipity of science, no doubt about it. Do any of these animal viruses pose threats to humans?
Well, we're not sure if there might be some noroviruses that are transmissible to humans. There have been some spillover cases with some bovine coronaviruses. Again, some of the sapoviruses are fairly similar to humans. The interesting component there for the norovirus and sapovirus that are in cattle and swine is that we did an epidemiologic study. We found these animal norovirus, calicivirus are also present in shellfish. Mm-hmm. So for the people who eat raw shellfish, not only are they eating human norovirus that comes through sewage, but also the animal caliciviruses. So that's a caveat if you like raw oysters.
Yeah, I would stay away from, they're great, but cook them. What's that?
Keep your immune system educated.
That's right.
A little bit of virus
you have a few papers where you're looking at noroviruses in lettuce, binding to lettuce, right? What is that about?
So that's in collaboration. I've been lucky to have tremendous postdocs, graduate students in my lab. So right now we're very fortunate that we were able to retain one of my very talented graduate students, Dr. Kian Wong. And she's actually taken over some of that work, and she's actually written a USDA grant for that. So we know that noroviruses bind to histoblood group antigens, which is a unique feature of noroviruses, and that allows them a big repertoire in humans for different histoblood group antigen types. So every blood type, there's an analogous norovirus that can bind. So we thought, well, maybe there's something going on in these uncooked vegetables that actually allow the virus to bind so you can't wash it away. So there are some lectin-like binding particles that the norovirus binds to lettuce.
Got it. So is there any one experiment over the years from your lab that you particularly like? I know it's a loaded question because it means you're going to single out someone's work, but sometimes there's one.
Well, I would have to say probably two of them that had the major impact was the passage of the human norovirus in the nodobite pig model and then being able to grow it. And that was a key for future research on that virus. I think the other one was this gut-memory connection, discovering that the cells that recover from the virus infection and the next litter, the litter can be protected, and that's due to this high level of Ig antibodies in the colostrum milk.
Okay. So let's go back to you, Margo. Now, I asked you before about one experiment, so let's look at it differently. What do you think has been your biggest gift to science, let's say? Your most important contribution to science as a field?
Probably cloning the OAS1B gene. I think that was probably, but the thing is now that we know what the gene is, we still don't know how it provides this amazing resistance. Right. So now that has led to studies of partners and looking at whether that complex is in the region where the replication complexes of the virus are located.
Okay. So, Luis, what do you think is your important contribution to the field?
Well, probably I will differentiate two things. To get a reverse genetic system for a coronavirus was
Can you
meant the development.
Poke the mic. There you go.
Probably one thing was the development of a reverse genetic system for coronavirus. This is a very important tool. Once you achieve this, you can modify the life of the virus and turn a pathogenic one in a non-pathogenic one, and analyze the relevance of the different genes. And certainly, other people did it immediately. Ralph Baric, using a completely different strategy, he also succeeded. And later on, deleting one by one different genes, we identify the small envelope gene, a small protein with many activities and many domains. Deleting a gene has been very important because this gene contains at least two domains involving the virulence. Young students, they think that a virus may kill because it grows to high titers, but certainly this is not the case. There are many viruses that they are growing in our body and they cause very little pathogenicity. The pathogenic viruses are those that they carry virulence genes, and by identifying those virulence motifs, this is very important because you can change the life of a virus. So working on these lines, we identify E protein as a protein containing virulence genes, and that has been highly productive in our life. So I think that was very important.
How about you, Linda? What's your contribution to science?
Well, I mentioned a couple of our what I thought were more breakthrough research, but I would have to say that I think for a professor, your most important contribution are the students and postdocs you train. And I think that's our legacy to science. That they continue in the area and continue to develop the field and the knowledge.
I always hope someone says that when I ask people that. So Margo, science is hard to do. It's hard to get jobs. It's hard to get funded. Do you have any advice? You've been in the field a long time. You've seen things change. Do you have any advice for the young people out there who want to be in science?
Well, I think a lot of luck is involved. And when, for instance, in my career, I didn't get to choose where I landed. It was based on family issues. And then I had to make do with where I was. But I found good collaborators, and I went where my experiments led me, and was never afraid to learn new techniques or jump into a different area. So I think that being able to really think broadly and yet think about what's relevant to what you want to do and how practical it is, whether you can really assemble what you need as far as people and techniques to get the job done. Those are all very important issues in succeeding.
Right. Luis, do you have any advice for young people?
Well, the advice will be just get in science only if you really like it. And your life in the lab should be, let's say, 50-50 in terms of working on the bench, and the other 50% reading a lot. Because getting the information is very important, and that's very helpful. Otherwise, it will not be so easy, novelty, to do novel things. So,
nice.
I think it's important.
How about you, Linda?
So I think probably important things are be passionate about what you do. I think that's what keeps you in science and keeps you working in the area. Other things, you probably need to realize funding is cyclic. And there'll be downsides and upsides, and I've seen the both. So when there are downsides, you need to be flexible. You need to think of other sources of funding to support your research and keep going.
All right, I have one question for you all, and that is, Margo, if you hadn't been a scientist, what would you have been?
Well, when I was a young woman, I thought about being a ballet dancer, and then next was an archaeologist, and then I was thinking about going to medical school, but really what changed my mind was my senior year in college. I washed dishes for a graduate student, and I sort of listened to her. There was no one else. She had a small cubicle that she worked in, and so it was just the two of us. And she'd tell me about, what she was hoping to find, and then if it worked, it was really exciting. And that, I never even applied to medical school.
That's a great story. We always tell people, they ask for advice, how do I get into science? We tell them, go wash dishes in a lab. So you're validating that. Luis, how about you?
Well, I remember that I wanted to be a teacher. So that was all my passion at that time. But then I also needed money, and to work. And then I got this job in the medicine faculty, which I enjoy very much. But if I will not be a researcher, I would like to be teaching different levels, because to teach properly, you need to understand what you are talking about. And it's the only way you can teach properly.
I think scientists are really teachers in a way anyway. That's how I view myself. Linda, how about you?
Well, actually, after the launch of Sputnik, during my childhood, when I was growing up, I really would have liked to have been an astronaut. But I got deadly carsick. And I thought, this is maybe not a good career to aim for.
That's funny you should say that, because the last TWiV I did in New Orleans at ASM Microbe was with an astronaut. Kate Rubins was a virologist, and she was working at the Whitehead, and she applied for the astronaut program and got in. And now she went up last year, and so check out the last TWiV 444. Stephanie, you can show her how to listen to it, okay? She should listen to that because she is awesome.
Oh, great.
And you should hear about pipetting in space, how cool it is. You'd think the liquid would fly all over, but it doesn't. The surface tension keeps it on the plastic. So she's done sequencing, she wants to do cell culture, and you know why she does this? Because they want to put a base on Mars, and they need a lab there in order to diagnose things when people get sick. They want to look at the microbiome in space. So listen, TWiV 444, Kate Rubins. She's just awesome. All right, we have 4 minutes left. I know you have another session, so I'll wrap this up. You can find all the episodes of TWiV at Apple Podcasts. You know, if you have a cell phone, there are apps that you can use to subscribe for free. Every episode you can get, or you can go to microbe.tv/TWiV. And if you have questions or comments, you can send them to TWiV at microbe.tv. And if you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have a Patreon account, a PayPal account, Amazon affiliates. We sell t-shirts, mugs, everything at cafepress.com/TWiV. I want to thank everyone for participating today from Georgia State University. Margo, Brinton, thank you so much.
Thank you.
From the Spanish National Center for Biotechnology, Luis Enjuanes, thank you so much, Luis.
Thank you.
From Iowa State University, Linda Saif, thank you so much.
Thank you.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the organizers of this meeting. Ying Fang, thank you for having me. Susan Baker, thank you for having me. Thanks to the audience. I'm sorry about the sound problems, but they fixed it. Thanks, guys. Thanks for the sound people. And I have 3 more t-shirts to give out here. Anybody want one? 1, 2. Hey, Luis, can you grab that shirt and give it to me? 1 more. Well.
Oh, wow.
Ralph, are you going to wear that shirt? No, it's either medium or large, so you could swap with someone else if you want to.
I'll squeeze in.
The introductory music on TWiV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.